But like in the announcement and in the instructions, it says do the first one. So now you have 7 additional practice problems that you can work for fun if you don't know if you're doing it right. 11 students got all 3 right, even though some people forgot units, so it did detract from some people's scores. 41% of students got it right, so the average was kind of 41, kind of ish. And then some people got 1, but not the other distances. Some people didn't get any. And one thing we need to look at is what you do. And so if you can't figure out what you did wrong based off of your paper and the answers, then talk to somebody else, class, come talk to me, bring it up on a Friday so we can go over these problems so you don't miss them. Unfortunately, the thing I really don't want to sort of be mad about is that people didn't follow my directions. 9 of you didn't put some centimorgans on your answers. Shame on you. Shame. Shame. You lost some points because you didn't put centimorgans. And for some reason, a lot of you decided to do more work than required. I'm not sure why people want to do more work than they want to do, but some people did. And I'm very surprised that people could do all 8 questions in 20 minutes. It takes me between 3 and 5 minutes to do 1 question. So one thing I want to keep in mind is sort of your time period when I try to shoot for it. Can I do this in 5 minutes? So I have time to do the Punnett square, binomial calculation, and answer all the other questions that are on the test. Or you could say, fuck it, I'm losing those 9 points, I'll fill my time elsewhere. Which is a fair strategy if you want to do that.
Freaky people.
But again, Friday is the day to ask questions, test on Monday, so if you still haven't figured out what you did wrong and if you're going across, bring it to class, we'll look at it. If you have it with you today, I can look at it a little time after class, look at it for group ball or agreement shows up. Not sure who it's going to be today. What do you think?
Greenwood.
Neither.
Greenwood. Greenball's not going to be here. He's already.
I guess he was on trip.
He's on trip.
The board, I was right in the front, just to remind you of how you approach the 3-point column, and these are things you can look for, trying to spot your mistake. You don't really know what you did wrong. And as we look at the steps, the first step is to identify the parents, which again should be the highest number. And there's going to be 2, so let's talk about it as being a pair. Same thing is true for step number 2. We identify the double recombinants, which would be the lowest pair. And now that we have these 2 things, we can do the next step, which is to identify the middle, which requires us to compare the parents to the doubles. And the whole idea of what we want to remember is in a 3-point cross, the middle gene is the gene that switches. So we should see the parents having 2 alleles in common with 1 double, the other parent having 2 alleles with the other, and one that's different is the middle. And then once you have this, you got to rewrite your things, rewrite parents in correct order. And this is a place where some people get things wrong for whatever reason. They use doubles to rewrite rather than parents. And then we'll do the first recombination, or single recombinant, or crossover. And so if you determine your order to be, say, A, B, C, then the first thing you do is do the recombination between A and B, which gives you the distance between A and B. This gives you another set of numbers, third set of numbers, third number pair. Once we have the third number pair, we can easily identify the other pair because it's the one left over, and that's going to be our other single crossing over event. But in doing the calculations, we don't make sure that we include in our numerator the number of singles plus the number of doubles divided by the total. And some of you may have gotten that wrong because you didn't divide or have 4 numbers divided by 8 numbers. which can change things. Like I said earlier, if you got the right numbers but not the right places, the most likely thing you didn't do correctly is rewrite your order in the new order. Because that is one way to get the numbers right, but not the positions correct. Double check you rewrote your parents as the right thing. And another way to sort people mistakes is they don't use all 8 numbers, they just divide by the number of parents. We're kind of used to doing that in other calculations. So take a look here. Hopefully you have your sheet still. You can look at what you did. Hopefully try to figure out what went wrong so we can get it right. On a quiz, 9 points, not gonna be that big of a deal because you're gonna get more than 100 points at the end of the semester. On an exam, 9 points is a bigger chunk of your grade. So this is a little more important. Try to get those 9 points. The question on the exam would be the same format, same style. Different letters, different numbers, different middle gene. But shouldn't be any surprises what it looks like and what you need to do with it. You just have to make sure that you know what to do with it. Same thing is true for the binomial Punnett square calculation problem. Same format that was on the previous quiz. Different numbers, different letters, different colors, who knows. Shouldn't be any surprises on what it looks like. What you're asked to do with it is just, can you do it? Any general questions about 3-point crosses? Can't spend too much time on it because we can just deal with that on Friday also. No general or any questions about 3-point crosses right now? Okay. Just to remind you, we are talking about replication, but more specifically, it's not necessarily how replication works, but how replication is kept error-proof. It can't be completely 100% error-proof. We're trying to create fidelity in the replication process. And what I like to think about is how much does each of these things contribute to the individual versus the total sort of error rate that we get. And there are 3 components that we need to think about. The first is just the DNA synthesis aspect. So we need more space of DNA polymerase. Want to remember what it is. These are the same. We get 10 to the minus 5. So we get a pretty good error rate just because of the way the enzyme works by itself. If we add in proofreading activity, which is also a component of DNA polymerase, which again is something I think is important to be able to separate, we get a little bit better, but still not as good as we could do. The reason why I like to think about it this way as individual and total is sometimes we get situations where proofreading activity is mutated, and we still sort of figure out how much proofreading or error proof or fidelity we have in the world where proofreading activity is not around. But what we need to talk about today is mismatch repair. And this is not something that the DNA polymerase does. There are a different set of proteins and enzymes involved in this process. But ultimately, we can get a total error rate of 10 to the minus 10 by including both proofreading activity and mismatch repair together. The proteins that are involved in this are called MUT, or mute, for mutations, or mismatch repair. I don't really know, but there's 3 of them. You've got H, you've got L, and S. Each of these is involved in a slightly different purpose. That's one of the things that we'll start to do this semester is learn what some individual proteins do. We already know one, hopefully DNA gyrase, protein we all can remember, an enzyme, big member, what it does. And one thing we left off on Wednesday, no, today's Wednesday, Monday, sorry, with mismatch repair is the importance of methylation. And we use methylation as a way to determine if our DNA is a new piece of DNA or an old piece of DNA. And that's going to introduce some terms we need to think about and some ideas. The first thing we're going to look at here on the slide is that there is a particular DNA sequence that is used as the methylation site in bacteria. That's an important distinction. Bacteria methylate DNA in a different position and different location than eukaryotic organisms will. So for bacteria, there's a 4 base pair sequence that has to be present for the DNA to be methylated. That's what we call GATC. It's in this 5' to 3' direction. So it has to be in this order, from left to right or right to left, doesn't matter. GATC, but it's also GATC backwards as well as forwards, because again, it's complementary. Of these 4 nucleotides, it is the A that will actually be methylated. But only adenines that are present in this GATC sequence are things that are methylated. What's the probability of getting these 4 nucleotides in this order? Do they have any idea how we would calculate that particular type of probability? Yeah. But is there one order or multiple orders? I just want one order. Okay. It has to be this sequence. So yes, it is just 1 to the 4th to the power of 4. So this is not going to be a very common sequence. Whatever 1 to the 4th, 1 over 4 is, so you know, 0.25 to the 4th is a pretty low number. But these are not very common, and they're spread out around the genome purposefully, so you don't have like too many of these out there. But there's some every once in a while. They're kind of like mile markers on highways. But you're every mile, because they're called mile markers. They're not like every feet or foot, because that'd be redundant. Who cares? You need to know every foot. You need to keep being identified as being what you are every second, or is it okay to know, like, I'm who I am on every hour or every day? It's okay. So we don't need a lot of these, but we do need a few. So we methylate the A of this GATC sequence. Now methylation is important for mismatch repair, but it also turns out that methylation is also a protection mechanism that bacteria use. By methylating their DNA, they mark it as different than other DNA. And typically, bacteria methylate their DNA to protect themselves from new foreign DNA coming in and causing problems. So if you introduce methylated or unmethylated DNA into a bacteria, it's automatically going to eat it, destroy it, because it says this is bad DNA, it's not my DNA. So unmethylated DNA can be targets for destruction. But during replication, there is going to be a place where methylation state is going to vary. And that's going to give us an idea that we need to think about called hemimethylated versus fully methylated. And when we start the replication process, both strands are methylated. They're considered to be whole pieces of DNA or parent DNA strands. And as we pull those two apart and make new DNA, the new DNA that we make is going to be unmethylated for a short period of time. This half methylated piece of DNA molecule is what we call hemimethylated. So hemi is half. Could have called it semi-methylated, but they call it hemimethylated for whatever reason. I don't know. Maybe it has something to do with trucks. Hemi, something like half or something. I don't know. But whatever, hemi is half. One strand of a double-stranded DNA molecule are methylated. And it's always going to be the parent strand that's methylated. The daughter strand, or the new piece of DNA, meaning the leading strand and the lagging strand we make, are going to be unmethylated for a short period of time. Proofreading activity works immediately. As the polymerase is making the leading strand, lagging strand, it checks to see did it add the right nucleotide or not. This system works after we finish the whole process and before we start the next round of replication. So how much time is there between finishing and starting the next round of cell division? Is how much time mismatch repair has to fix anything that's gone incorrectly. And it's called a mismatch repair because we do get a mismatch in the DNA sequence. One nucleotide is paired incorrectly with a different nucleotide. And so in this idea here, we have our daughter pieces, parent DNA shown here in black. You can see the A showing the methylated site, or the methylated nucleotide, and the methylation site of GATC. So you always get two A's methylated sort of right near each other in our parent old piece of DNA. Again, through replication, we form a bubble which pulls apart the DNA. We put leading strand and lagging strand against that old piece, so we get our new piece of DNA. So here I've highlighted a mismatch. C and T are not supposed to go together. But over here we were okay. We got the right nucleotide base paired, so this piece of DNA is fine. This piece of DNA has got an issue. If both strands are methylated, there's a coin flip on what you change. So because only one strand is methylated, the repair process knows that this is the correct piece of DNA. This is the wrong piece of DNA, so this is what should get fixed. Proofing activity is very surgical. You only remove one, because that's the only thing that's wrong. Unfortunately, mismatch repair is not surgical at all. It's bomb the hell out of it and fix it later. Because what we do in this process is to cut the strand with the wrong nucleotide, and several hundred of nucleotides are removed at once, only to be replaced later on. That's like fixing a typo by rewriting the entire paragraph instead of just finding the one mistake. Or let's fix a pothole by taking up the entire mile of road and repaving the whole thing to fix one little pothole. But this is an easy process for the cell to do. It's very quick and easy. You might think it would be easier just to remove one. But for the cell, it's quicker to remove a bunch of DNA and not worry about which one was actually wrong, but to know that there is something wrong between here and here, take it all out and replace it all. It's just an easier, more simplistic idea for the cell, because it can't spend all its time looking for where is the mistake. We know there's a mistake somewhere, but it's somewhere between point A and point B. Two of the proteins, MUT-S and MUT-L, are important in finding the methylated DNA strand and identifying that there's a problem here. We're not going to separate what MUT-S and MUT-L do, because they're both involved in this. Basically, let's find the mistake. When it comes to trying to fix the mistake, the activity of MUT-H is something that we will want to remember, because MUT-H is a DNA cutting enzyme, just like DNA gyrase is a DNA cutting enzyme. Just like transposase is also a DNA cutting enzyme. DNA gyrase cut both strands on one location. MUT-H is going to cut one strand in two different locations. We basically need to cut out this bad part of section. And then once it's gone, we can replace it. It's like any type of surgery where you're trying to fix a ligament. You pull out the whole thing and you put a new one in from somewhere else. Well, here we're not getting it from somewhere else. We're actually remaking it using DNA polymerases and other things. And one of the cool things about this mechanism is really no different than the primer removal and primer replacement mechanism. It's just applied in a slightly different fashion. But it does require half methylated DNA for it to actually work. If the DNA is fully methylated, we have no idea which is the right nucleotide and which is the wrong nucleotide. So half the time we get it right, we fix it, the other half we get it wrong. And if you want to use some important scientific language, we could call MUT-H an endonuclease rather than an exonuclease, because it doesn't cut at the end of the DNA strand, it cuts somewhere in the middle of the DNA strand. And to reinforce the idea that it's only cutting one strand, we call it a single-stranded DNA endonuclease. And for the most part, it's considered sequence independent, because it's not going to bind to a specific DNA sequence. It's going to be able to recognize all types of DNA to cut things. Now again, this was something that was originally found in bacteria, but it's also present in eukaryotic organisms like us. Humans, for instance, do have a mismatch repair system. we find homologs of mute L's and mute S's. There's no homolog of mute H. So if you remember mute L and mute S allows us to find the mistake, mute H allows us to fix it, there's some piece of the puzzle missing here. The other thing that's weird about eukaryotic organisms is our DNA is not methylated at a GATC site. Eukaryotic organisms like us will methylate GC or CG sites. This particular fact is not important right now. You can forget about it, because it'll be much more important for exam 3. I don't want you to overburden yourself with things that are really not important right now. This will become more of a story as we go forward. But just in the back of your head, if you remember that methylation of DNA is sort of universal, but how it's methylated is going to be different. And the purpose of methylation is also different in eukaryotic organisms. Which is also why I don't want you to worry about eukaryotic methylation right now, because it's not involved in mismatch repair, as far as we know. And again, there's a small window where mismatch repair can function. As soon as the DNA becomes fully methylated again, mismatch repair is really not going to be helpful. And 1 out of 2 is not bad, right? Flipping the coin, 1 out of 2 is not bad. But still, would you take that chance? It's life or death. That's sort of what the cell is doing when it's trying to fix DNA. When we talked about meiosis, and specifically non-disjunction, one sort of weird gender effect was that in oocyte production, non-disjunction is going to be more and more common as the woman gets older. And non-disjunction really, rates don't really change in males' sperm production over their lifespan. Where we do see a difference, sort of gender difference between men and women, is that replication will be a greater contributor to mutations in men than in women as age increases. And a lot of this has to do to the fact that DNA has already been replicated before you're born, basically, as all those cells are sort of arrested in meiosis at some point, somewhere in this prophase area. Not to memorize that, but we've already replicated our DNA, so we copy our DNA with new, fresh enzymes, so there should be no mistakes. But as men get older, our DNA polymerases get worse, and more mutations get in there. Proofreading activity is not as good. Mismatch repair is not as good. More mutations become possible as we get older. It's one of the reasons why it is important to take age into consideration when thinking about having children. The older you are, the more chance there are for mistakes to happen or to be already present. One of the cool things that came about by learning about replication was DNA sequencing technology. There's a direct link between understanding how cells make DNA to how we can use this outside the cell to essentially make DNA. And Frederick Sanger is the developer of this sequencing process. It's called the Sanger sequencing method in honor of him. And the way it works is we use particular molecules called dideoxynucleotides. And as you can see from this slide here, what is a dideoxynucleotide? It's something that doesn't have a 3' hydroxyl group. This is a regular deoxyribonucleotide. This is a dideoxyribonucleotide. Without that 3' hydroxyl, there's no way to add a second nucleotide or another nucleotide to the chain. So these dideoxynucleotides are called chain terminators, because once they're incorporated, they stop the replication process. And that might not seem very useful by itself, but we can add things to it that'll allow us to see the DNA sequence. The old school method was to use radioactivity. And this is an example of what a original DNA sequencing gel would look like. You have a radioactive A in one reaction, a radioactive G in another. And you have 4 different reactions where you add these dideoxynucleotides. And they make a DNA molecule up to a certain point and then stop. You basically read this like a sequence from bottom to top. Not you need to remember this, but it's a little confusing because you gotta figure out like where things are. It works pretty well. You just kind of gotta read up, you know, where you are, and that tells you which letter you are based on where you show up. The reactivity is bad and sort of hard to deal with. And also you have to do 4 different reactions. So somebody came with the idea that we can add colors for rest of molecules to the nucleotides. And this is what modern sequencing will do, is every nucleotide is a different color. And we get a readout that's called a chromatogram, which we'll see here in a second. And it's much more user-friendly in seeing the DNA sequence. One of the other really important things that has happened over time is that the price to sequence has really gone down quite a bit. Even in the sort of late 90s, early 2000s, you could publish a paper by sequencing a genome, because it was actually a big deal back then. But now, you sequence genomes in an hour, almost. That's probably hyperbole. But it's really quick, really easy. And this is what our modern day sequencing reactions look like. We get all these peaks and valleys, and each peak of a different color represents a different nucleotide. You don't really need to memorize this other than to analyze or to identify the DNA sequence. We get this thing called a chromatogram, which is a color representation of the nucleotides in a particular order. Has anybody done 23andMe or Ancestry.com, or what kind of pet do I have? Did you know you can sequence your pets to figure out what kind of breeds are actually in your pet? Pretty important if you're worried about diseases, because some breeds are more prone to certain types of disorders. But when you send in your sample and they do sequencing, this is the type of readout they're going to get. So DNA sequencing did directly come as an offshoot of studying the replication process. The other thing that came from replication was the process called PCR, or polymerase chain reaction. PCR was actually needed to do sequencing, at least nowadays, but before, or at least initially, they were 2 separate things. The guy that did this or developed this was Kerry Mullis. Here's him. I don't know why he likes to have a picture of him naked, or half naked, with a surfboard on the beach. But if you read his book, and you can read his book, it's in the library, he talks about going on a trip, not an actual trip, but a thought trip, induced by mushrooms, and marijuana, and other herbs, and other psychedelic entanglements. And he tripped up the idea of maybe we can make DNA outside of the cell. And he made it happen, which is really cool. So I'm not saying that drugs are good, because drugs are bad, okay? But at least at this point, it was able to sort of free his mind and think about, yeah, maybe we could do it. And so PCR, really important in every sort of laboratory setting. And you can't go anywhere without talking about PCR. But it's a way that we can make DNA outside the cell. It's not foolproof. We can't make large pieces or really big pieces of DNA. But we can make some fairly, let's call them medium, small, medium, I don't know. Medium size. As we look at the steps involved in the PCR process, there are repeating the step of, or process of 3 different steps that go in order: denaturation, annealing, and extension. These 3 things mirror the first 3 steps of the replication process. Denaturation refers to taking a double-stranded DNA molecule and heating it up to the point where they can't hold themselves together any longer. That's what bubble formation does. A bubble formation is enzymatic, so it can happen at any temperature. If we're doing it mechanically, we have to use temperature to sort of break these things apart. Once we break them apart, we need a primer, at least 2 primers, to make the DNA molecule that we want to make. And that's where the annealing step comes in, because the primer has to base pair to the DNA sequence. And once that happens, a DNA polymerase can be used to extend that primer to make a piece of DNA. And after time, you get a product that is the right size for what you want. Here, replication requires a primer, requires a DNA polymerase, requires bubble formation. What really made PCR actually possible was the identification of DNA polymerases that work at really high temperature. So to do this process, you need to be able to break the DNA, and that requires a fairly high temperature for this to happen. And using the power of math, you get an exponential increase in your product over time. So the more time you do it, the more product you actually get, and you can get something that's actually usable for some molecular biology tool or diagnostic. So those things we get from the STR gels are most likely made using a PCR reaction, specifically for certain parts of the DNA sequence, so to read into these places where there's lots of repeats. So make the product and then run it on gel and see how big it is the cool thing about PCR. It makes the thing that we can then analyze later on. And PCR was a pretty cool thing, but after this first type of process, there are, or were, two different sort of developments or modifications of the PCR process. The first up here is called RT-PCR, for reverse transcriptase, because it goes backwards. PCR uses DNA to make more DNA. RT-PCR uses RNA to make DNA. Sometimes needed because some organisms don't have DNA genomes, like viruses. Also for humans, there's a lot of bullshit in our DNA sequence that is not going to be present in our proteins. And so a lot of times when we try to sequence a gene in a eukaryotic organism, we target the RNA rather than the DNA sequence, because the RNA is not going to have any introns in it. So you can get the full length gene sequence rather than have to worry about the introns and the exons and all that nonsense. Now, one of the cool other things that happened because of PCR was something called qPCR, which stands for quantitative. Now, sometimes qPCR is also called real-time PCR, which is RT-PCR, which can be confusing because we don't want to confuse real-time PCR with reverse transcriptase PCR. So we'll use qPCR instead, and RT-PCR refers to this specifically. But this allows us to measure gene expression by seeing how many RNAs are actually being produced or have been produced over time. And as we go forward, for exam 3, there'll be a few more things we'll talk about that allow us to measure gene expression. It's not just what is your alleles, are they dominant or recessive? It also matters how much of something are you making, because that can also have an impact on your phenotype. Ta-da! As they say. If I was really good at it, I would do my own walkout. But I'm not using, I can't really do this thing. I can do this. Here, that's not what I kind of do. We got 16 minutes. Does anybody want to talk about anything before Friday? I'm here until everybody else is gone. So if you want to talk about your 3-point cross or anything else, feel free to do so. We've got now 15 minutes of extra class time available. I know some of you probably want to spend more time with me than you have to. So feel free to get up and leave. Will not hurt my feelings.